Welcome to the third session in the Cell Biology Cellular Organization Division and Processes course. Um, I am Smriti. I am a PMRF TA who has been allotted to this course. So, while the PPT is on, I request one of you to admit people in the lobby because then when the presentation is on, I will not be able to see that. So, as I was saying, this is a session 3 for the NPTEL course. I am Smriti. I'm a PMRF TA who's been allotted to this course. Such sessions is basically meant for discussion and questions, and if you have any doubt, and we can clear that as well. This will happen thrice a week by 3 different TAs. One is on Saturday, one is on Monday, and one is on Tuesday. You can join any one session based on your convenience. And this session, which I am conducting, will happen every Monday from 6 to 8. So, in the last few sessions, we have discussed about the week 1 and week 2 of this course, and we've discussed the recent discoveries and a few techniques that are used in the lab to study them as well. So, this week, the session 3, which is week 3 course, basically have this content known as DNA replication and its importance. So today also we will discuss the recent discoveries and talk about a few techniques that were used in the lab and discuss a few landmark papers that first initially helped establish the field. So to begin with, what we saw in this week was that all cells have to move into the S phase only after it crosses a checkpoint. This checkpoint, often called a start or restriction point, is a point that is mitogen dependent. Only if the cell has favorable condition does it enter the S phase and replicates its DNA, because DNA replication and cell division in general is a very large energy consuming process. Hence, only if enough nutrients are available and appropriate growth signals are present, the cell will initiate this process. Now, once the cell crosses this S point or the R point that has been labeled here, it enters into an irreversible phase wherein the cell cycle will progress, and there is no way the cell can come back into its senescent phase. So, how this basically happens is, if the appropriate growth signals are present, then these signals will initiate the transcription of the cyclin gene. The cyclin D will then be transported into the nucleus, and here it will form a complex with the CDK4, and this active cyclin and CDK complex will further process to do the downstream activities, including phosphorylation of this protein known as RB, which releases the transcription factor E2F. This E2F will then proceed to translate cyclin E that will help the cell move into the S phase, which is where DNA replication happens. And it also helps phosphorylate certain inhibitors such as P27, which generally stalls the CDK activity. I would also like to say, if any of you have any questions, then do unmute yourself and interrupt me anytime in between. That will be more convenient. So, as we know, right from prokaryote such as a bacteria to a eukaryote such as a mammalian cell, all these cells have this blueprint known as the DNA. This DNA, whose structure was found to be a double helix structure, generally has all the information that is required to synthesize all the proteins that is present in the cell. And it also is organized in a manner, chromatin in prokaryotes and in chromosomes within eukaryotes. And this has to be replicated without any error, because once an error happens in DNA replication, then the daughter cell will inherit this mutated form, and that could have catastrophic effects. So DNA replication needs to be highly accurate, and a lot of measures have been taken care of by evolution, and today DNA replication by the enzyme DNA polymerase is very, very, very accurate. So basically what happens is that there is this multi-protein subunit, and there is DNA helix which unwinds the parental DNA helix, forming two single-stranded regions. The single-stranded region is bound by these single-stranded binding protein that stabilizes the single strand and does not allow any endonuclease attack or destruction of this open DNA. There are these sliding clamps and clamp loaders which are present, and they essentially slide onto the parental strand, and this prevents DNA polymerase from falling off, which means that it can synthesize a long region without falling off the DNA. And there is all, of course, DNA polymerase present, and this is the actual enzyme that has the catalytic activity that is able to add DNTPs onto the 3' end of the strand, and a new strand of DNA is synthesized. In this case, the parental DNA serves as the template, and a new strand is synthesized based on that. So, because the DNA polymerase can only add nucleotides to the 3' end, there is a leading strand and a lagging strand. The leading strand is synthesized from 5' to 3' in a continuous fashion, and the lagging strand, on the other hand, has to be uncoiled, and then primers are added, to which small fragments of DNA are synthesized that are later joined together by the ligase enzyme. These fragments, because it was identified by a scientist named Okazaki, it's called Okazaki fragments. And this is later joined together by DNA ligase to form one complete strand of DNA. So, like I told you, DNA replication has to be very, very accurate. And this is one of the reasons as to why it is so complicated, as in why there are Okazaki fragments produced, why there is only 3' addition, why there is an RNA primer, when simply there could be a DNA primer that is attached, and DNA primer could later be joined together by the ligase. But currently what happens is there is an enzyme that adds an RNA primer that is later degraded and then refilled by the DNA polymerase to form the final daughter strand. And this is because if an enzyme is capable of adding strands de novo to a template, as in if it is not just extending, if it is capable of adding the first nucleotide to the template, then it is known that it is often inaccurate. So imagine that there was a DNA primer that was added, but this primer has to be added by an enzyme that is adding the bases de novo, because of which there is a large possibility that these primers are inaccurate. So in order to avoid that, the system ensures that RNA primers are added, which may or may not be inaccurate, but they are removed and finally sealed together by the DNA polymerase enzyme only, so as to maintain the accuracy. Another activity of the DNA polymerase in itself is this 3' to 5' exonuclease activity, which serves as a proofreading. So essentially, say there is an incorrect base that is added, because the only activity of the DNA polymerase is to choose bases and add it to the 3' end, and any base can be added. Now imagine instead of T over here, a C base is added. Now the structure of the DNA polymerase is such that it is not able to elongate bases that are not accurately base paired. So because the C is not base paired properly, its orientation is such that it does not approach the catalytic center at all, and the enzyme is not able to add the next base to it. So it is stalled, and then there is this 3' to 5' exonuclease activity, which will cut and remove this inaccurate base so that an accurate base can then be added, and only then will DNA replication proceed. So these two are two of the basic mechanism by which DNA replication's accuracy is maintained. There are, of course, other mechanisms as well, which we can discuss later. So now we'll move on to the previous year assignment questions, and we can solve them together and discuss a few basic concepts behind these questions. And then we'll move on to the techniques and paper discussion. So the first question was, pick the correct statement about S-phase checkpoint. The S-phase checkpoint doesn't respond to DNA damage and replication perturbation. a defective S-phase checkpoint causes irreversible replication fork collapse. The S-phase checkpoint allows the continuation of the cell cycle despite destabilized replication fork. So the S phase, as we know, is the phase in which DNA replication proceeds. So what this checkpoint basically does is it's present in between the S phase, and here it checks whether the DNA has been replicated correctly, and only then will it allow the cell to move to mitosis. This again is done by a large number of proteins which are inhibiting each other or phosphorylating each other and ensuring that the next phase does not start unless and until DNA has been replicated correctly. So out of these options, if we see that the S-phase checkpoint indeed responds to DNA damage, the S-phase checkpoint only allows the does not allow the continuation of cell cycle if it has destabilized replication fork. So this is also incorrect. And the first statement is also incorrect. And hence the correct statement would be that the defective S-phase checkpoint causes irreversible replication fork collapse and then leads to genomic instability. So this is why S-phase checkpoint is considered to be one of the most important checkpoints to ensure that the cell is not having some catastrophic defects. The next question was, which of the following events takes place in a cell when replication fork hits a DNA lesion? A DNA lesion is basically, say, an incorrect base pair or a strand breakage or any damage present in the DNA. And this will initiate these repair proteins. And the one that is in yeast, the one that is recruited is this MEC1 and RAD53. MEC1 is an upstream kinase that is able to phosphorylate RAD53. RAD53 then stabilizes the fork and does not let it move forward, and also inhibits mitosis, as in it inhibits the progression of cell cycle. And this is what this question is talking about. In S. cerevisiae, MEC1 gets recruited. In S. cerevisiae, RAD53 gets activated via phosphorylation. And the helicases and polymerases will leave the replication fork so as to expose the regions of single-stranded DNA that can later be accessed by these DNA repair proteins that will come in correct whatever error was in the first place. So the answer to this question would be all of the above. The next question was, in which stage of the cell cycle does licensing of replication origins occur? So licensing of replication origin, what does that mean? So we have seen that there are these regions in the DNA where the replication can start. These regions are known as the origin of replication. And here there is this protein called ORC, origin recognition complex, that binds to. To this, there are other proteins such as CDC6, MCM helicases that come and occupy this region to form something known as the pre-replicative complex. Now, this happens in the G1 phase of the cell cycle in itself. However, it does not start replication. Now, once the checkpoint proteins come, they check that, okay, everything is fine, the cell is of appropriate size, mitogens are present. Then this pre-replicative complex will be removed, as in the MCM helicase is phosphorylated and the ORC is phosphorylated, because of which they leave the origin, and DNA polymerase is recruited to this place instead, and this happens in the S phase. However, the licensing, as in the region, as in identification of the region wherein the replication will start from, happens in the G1 phase itself. So it can start in the late M phase, which is basically the phase preceding G1, or happens in the G1 phase. Next question was about picking the correct statement about DNA replication. So we'll again go back to the image that we initially discussed. Here, the options were whether the DNA replication is semi-conservative. What they basically mean is if the daughter strand receives one parental DNA and one newly synthesized DNA strand, then it is known as semi-conservative model of replication. And that is what is happening according to the model in cells that we've identified. The other options were that in replication process, DNA strands are used as templates for the formation of complementary DNA. Yes, if you can see here, the parental strand is what is being used as the template for the synthesis of new nucleotides. At the replication fork, the DNA of both new daughter strands is synthesized by a multi-enzyme complex that contains the DNA polymerase. Indeed, there are a large number of enzymes that are playing a role. You need helicases, you need enzymes that are the primer, you need the polymerases. So all of the above, all these statements are correct regarding DNA replication. The next question was to pick the incorrect statement about Okazaki fragment. Okazaki fragments we saw previously were fragments that were present in the lagging strand. So these fragments are synthesized by the DNA polymerase in itself, but because it can add only in a 5' to 3' direction in the lagging strand, small fragments of DNA are synthesized, and this is later sealed by the DNA ligase. So the correct option could be that they are small fragments of nucleotide sequences. Yes, they're produced by the cleavage of DNA by endonucleases. This is wrong because they're synthesized in that way. It is not because of any cleavage. They are indeed synthesized in the 5' to 3' direction, and they're later joined together to create the final DNA chain. The next question was, what is a DNA ligase? So we have seen time and again that DNA ligase is the enzyme that is capable of joining the breaks in the DNA synthesized. So if there are bases added, but there is no joining between these bases, then that is where the DNA ligase comes into picture. But what bond does it actually form? So here is a representative of a nucleotide chain. So there are bases. The bases have a phosphate group at the 5' end, which is attached to the next base. And the base also has a 3' OH that is attached to the next 5' phosphate. And this is the linkage that is being formed. So that is why it's called 5' to 3' direction or 3' to 5' direction. So here we can see that in a final bond, to one end is one ester bond, and to the other base where a phosphate is attached to the OH group, there is again an ester bond, and in between there is a phospho. And that is why this bond is known as phosphodiester bond. So this is the bond that DNA ligase is actually capable of forming. It removes this, there's an OH and there is a O group over here. It removes this OH group. Actually, there is this OH group here as well. Here it has been represented as in the ionic form. Otherwise, there's a OH over here and a OH over here. What happens is this H and OH leaves as water and the bond is formed, and ester bond is basically formed. And so the answer to this question would be that it seals a broken phosphodiester bond. The next question was to pick the incorrect statement about a replication fork. So replication fork is an active replication zone progressively moving along the replicating DNA molecule. This is true. It structurally resembles the letter Y. This is also true because if you see that this is the parental strand, and because single-stranded regions are formed, this resembles a letter Y, and the parental strand is used as the template to form the daughter strand. So the incorrect statement here would be that it is present only near the replication origin because it keeps moving as and when the replication is moving forward. Even though it originates at the replication origin, it will progressively move along the replicating DNA molecule. The next question was to pick the correct statement about the single strand DNA binding protein, SSB. So we have seen previously that SSB are these proteins that bind to the single stranded region. And basically what they do is to protect the single stranded region, because otherwise they would initiate a lot of DNA damage responses, and the strands can get cleaved or it can form these structures. A lot of things can happen in the cell. So the function is to destabilize the helix, because the cell needs to open up the DNA in order to synthesize the new strand. They are unable to open a long DNA double helix. Yes, you need special enzymes. You need helicases to do that. They facilitate the DNA synthesis process by inhibiting formation of short hairpin helices on the single-stranded DNA. This is also true because if there is, say, a hairpin formation because of, say, complementarity in the single-stranded region, then the DNA polymerase enzyme will get stalled. It will not be able to move forward, and hence it is required that single-stranded binding proteins are present and prevents these secondary structure formation so that the polymerase can keep on synthesizing new strands. The next question was, which of the following is not required for DNA replication? The polymerase enzyme we have seen is what has the catalytic core to add the fresh DNTPs to the 3' end. DNA helicase is also required because that is what unwinds the DNA. DNA topoisomerase is an enzyme that prevents the supercoiling of the DNA at the region that is in front of the um, helicase. So if you imagine that there is this DNA present and there is a thread present which is already wound, but in one end you keep unwinding it, then you can imagine that towards the other end there is more winding of the thread, and that is known as supercoiling. And DNA topoisomerase is required, otherwise the supercoiled DNA might break or have other damages. DNA primase is again the enzyme that adds the primer, the RNA primer that is required because the DNA polymerase cannot start the replication. It can only add nucleotides to a previously existing primer. However, DNA methyltransferase is an enzyme that is capable of transferring methyl groups that is necessary in prokaryotes, which use this methyl group as an indication to whether it's an old parental strand or a new daughter strand. And hence that is not required for DNA replication as such. The final question was to pick the incorrect statement about DNA topoisomerases. They are reversible nucleases. They essentially break a phosphodiester bond. The topoisomerase 1 produces a single strand break, and the topoisomerase 2 forms a double strand break. And the topoisomerase are unable to separate to interlock DNA circles. So if you see, like I mentioned, if topoisomerase enzyme is not present, the DNA in front of the helix will have supercoiling and will have stress building upon it. And because of that, there are these enzymes known as topoisomerase 1 or 2 based on how they act. Basically, the topoisomerase 1 can cause a single stranded break. And now these two fragments of DNA can rotate independent of each other, because of which the supercoiling of the DNA is removed. In topoisomerase 2, what it basically does is it's able to cause a double stranded break, and if two circles are wound onto each other, both the strands will break and the circles will be uncoiled. So the incorrect statement would be that the topoisomerase are unable to separate two interlocked DNA circles because they can separate two interlocked DNA circles. Topoisomerase 2 enzyme is what plays a role in that. So with this, we are done with the previous year assignment question. I will move to the techniques that we use to study DNA replication, starting from briefly discussing Okazaki's paper and what he used to first identify that fragments are being formed. Before that, do any of you have any concerns that we should discuss before I start? Yes, this video will be uploaded in YouTube. The link will be shared to you. I don't know how they are sharing the link, but we have uploaded it on YouTube. The link will be sent to you shortly. Okay, then I'll continue. Do unmute and ask questions in between, because otherwise I will not be able to realize. So as you can see here, this is Okazaki's paper. This paper was published in the year 1969. The paper was titled Mechanism of DNA Chain Growth, and they studied this T4 phage, which is a bacteriophage that is able to infect E. coli, and it is a double-stranded DNA virus. So this is a very old paper, so the techniques that they use are very basic and very primitive in nature. So there is a little background that we'll discuss before we get into the actual figure of the paper. So before studying this synthesis of DNA, they had two observations. They observed that the polymerase enzyme is capable of adding these bases only to the 3' OH that is free. They realized that this enzyme cannot add the bases to the 5' end of the strand. So, like I discussed previously, this say is a chain of deoxyribonucleotide. Here we can see that in the 5' end there is this phosphate group, and in the 3' end there is this OH group. The researchers observed that to the 5' end, the enzyme will not be able to add any bases. So if you see, is they knew by then that the strand has to be separated and it is used as a template for the synthesis of two daughter strands. But if you see, when the strand is opened up, there is one 5' to 3' and one 3' to 5'. So if the DNA synthesis happens here near the fork in one strand, which is the 3' to 5', the synthesis can easily occur from in the 5' to 3' direction, which means that the enzyme can keep on adding the deoxyribonucleotides to the 3' end. However, in the other strand, 5' to 3', the synthesis should have happened from the 3' to 5' direction, which means that the enzyme will have to add the bases to the 5' end. However, they observed that this is not possible. This they did by studying the enzyme's activity in vitro conditions. Now, the second observation they had was that short segments of DNA accumulated in temperature-sensitive mutants for ligases. So, in the last session, we discussed how temperature-sensitive mutants were isolated. So, in this case, if there is a mutant for ligase present, then the Okazaki fragments that we now know today will not be joined together. So, in this case, from these cells, you can isolate these short segments of DNA, and that is what they observed. So based on this, they actually set up a very simple experiment. What they did was they labeled the strands, the newly synthesized strands, with a heavier isotope of carbon or a heavier isotope of hydrogen. That is basically to just visualize it. So these will be radioactive, and you can visualize these strands by autoradiography. Otherwise, in those days, DNA visualization was difficult. So they isolated the two different strands of the fudge. So there is this W strand, which is say one strand, and there is this C strand, which is the other strand. They are both the parental strands. And they saw that these short fragments that was accumulated, when you add them to this purified W or C strand, there was a short amount of annealing present. Annealing meaning that they could bind to this purified W strand, which meant that there was some complementarity. So they found that this short segments of DNA was complementary to both the W strand and the C strand. So it was complementary to both the parental strands of fudge. This is what they identified. So in a fudge, the type of replication that happens is different, and it is now known to be a multi-fork mode of replication, because of which they saw that both the strands are being synthesized in a discontinuous manner. And they had two conclusions, fairly simple conclusion. One is that the discontinuity of both daughter strands arises from a discontinuous synthesis along the two template strands. These fragments were, since this paper, known to be Okazaki fragments. And this implies that there is a multi-fork model of DNA replication. There's many forks that are initiated at multiple points in the template, and there is discontinuity in both the daughter strands. There was another hypothesis. This is because of endonucleotic cleavage and not because of discontinuous synthesis. But after the synthesis, there is some enzyme that is chopping up these fragments. But then they realized that this was not likely, because the labeling was sensitive to a 3' exonuclease. And it was seen that these fragments, because labeling only labels the newly synthesized bases and not the old bases, they realized that because they are sensitive to 3' exonuclease, which means that if you add 3' exonuclease, the labeling is lost from the cell, because it cleaves the last few newly added bases. Because of this, they concluded that it is not endonucleotic cleavage, but instead it is freshly synthesized DNA that is being synthesized in this discontinuous manner. However, this was based on a bacteriophage, and Soonly after, there was this paper in 1970 in the PNA's journal, which using a very similar experimental model by labeling the newly added bases, they showed DNA replication and in E. coli. And this is where they showed that there is one leading strand and one lagging strand, and the lagging strand is has discontinuity, which is now known as the Okazaki fragment. So all of these experiments basically require us to isolate the DNA. DNA, as we've seen, is present within the cell membrane, it is present within the nucleus, and there are a lot of other proteins along with it. There are histones attached along to it. There might be RNAs that are being transcribed that could purify along with it. And however, for these DNA replication studies, you need very pure DNA. So this technique was established fairly early on in biology research, in which any tissue is taken and the cell is lysed. The cell can be lysed by chemical means, as in you add any detergent. It can also be lysed by physical means, wherein you manually grind or homogenize the samples. And later on, you wash them. These detergents will help remove the membrane lipids, and they will leave along with the detergents, and you keep on washing. And finally, you add proteases to remove the protein contamination, you add RNases to remove the RNA contamination, and ultimately you get this purified DNA that you can use for your experiments. Now, the thing is that this experiment is fairly simple and can be done without much of the chemical requirement. So I simplified this, and this is basically something we can all do at home to isolate DNA from, say, a buccal cells. So all you need to do is to gargle with water and salt, and this will dislodge the cells from your mouth and the inner lining of the cheeks. And these cells will also contain DNA from which we can isolate it. We need to add a detergent.
मैम आपका म्यूट हो गया है.
Hello.
Okay.
Okay. So what I was saying was that you add dish soap to the solution and finally mix the solution. Then slowly you add rubbing alcohol. Rubbing alcohol is basically isopropanol that we will get in any medical store or any hardware store. This will change the dielectric constant and will ensure that the DNA is precipitated. So this DNA will form thread-like structures that we can loop out using a toothpick. And this thread-like structure will have your own DNA. So this is a fairly simple method that anyone can do at home and isolate a DNA. It will be a fun game for young people. Then we will move on to discuss this experiment that was conducted by Messelson and Stahl that we've all studied right from school. In fact, this experiment is known as the most beautiful experiment in biology because it is a very simple, elegant experiment that conclusively proved the model of DNA replication. This is again a paper that is very early on in biology research. It was published in 1958. And this paper was, the experiments in paper was performed by the two scientists, and I have attached a picture of these two scientists relatively recently in the year 2020 in an interview. And their story is very interesting because they were not from the same institute. They met in another institute over a course. And then they came back to Caltech in California, and they were working in two different labs on their own independent project. And as a side project started to evaluate this because they had a very interesting idea. So in those days, there were three models for DNA replication. One which is conservative, wherein the parent strand remains intact, goes to one daughter cell, and a completely newly synthesized strand goes to the daughter cell, which has been represented here. Here we can see that the parent strand goes, represented in purple, goes to one cell, and the newly synthesized strand represented in blue goes to one cell. So this keeps on happening, as in every cycle of division, there is one parent strand and one newly synthesized strand. There were a few concerns with this model. The major concern was, how does the machinery read and copy the nucleotide sequence? Because it is present within the helix. If we remember correctly, then the bases are present within the helix, deeply buried within the helix. And how does this machinery accurately read and copy these sequences was unknown. The second method was a dispersive replication model, wherein the parental strand is cleaved at every turn, and this region serves as a template and a newly synthesized strand is formed. So herein we can see that the final product will be that there are short fragments of newly synthesized strand, short fragments of parental strand, short fragments of newly synthesized strands, and short fragment of parental strand in both the daughter cells. Again, there was this concern that if in every turn of the DNA there is a cleavage, then how does the cell know in which order to reassemble these fragments? Now, finally, there was this semi-conservative model of DNA replication that we now know to be true. Here, what happens is there is one parent strand, it opens up, one becomes the daughter, it serves as a template for the daughter strand, and similarly, the other opened up fragment serves as a template for another daughter strand. So in this case, in both cells, there will be one parent strand and one newly synthesized strand. But again, in this model also, there were concerns because DNA is such a long sequence. How does this DNA unwind and how does it become single-stranded? During this process of unwinding, how is it possible that they do not get tangled with each other? So to solve this, Messelson and Stahl, they designed this technique known as equilibrium density gradient centrifugation. So centrifugation is a fairly simple concept. It says that if you rotate any object with a fixed length around an axis, then there is a force that acts on this object called the centrifugal force that acts away from the center, which means that if there is this tube which is a fixed axis and you rotate it around the center, then there is a force that is acting towards the bottom of the tube. So anything that is present in the tube will get to the bottom. So they observe that if you add this chemical known as cesium chloride and you spin them at very high speeds, 45,000 RPM, which means 45,000 rotations per minute, and you spin it for a really long time. Here they spun it around for 20 hours, which is almost an entire day. They observe that there is this nice gradient that is formed, which means that higher concentration of cesium chloride, higher density of cesium chloride is present towards the bottom because of more force, and lower density of cesium chloride is present towards the top. Now, again, there's this other concept in physics regarding buoyancy and density, wherein we know that any object has a buoyant force that is acting on it, because of which it will go to the region which is nearing its density. So if there is a heavier DNA molecule, it will go towards the bottom of the tube. And if it's a lighter DNA molecule, it will stay towards the top of the tube because that is the region which matches its density. So this is what happens in the experiment. And what they did was fairly simple. They took an E. coli cell and for a very short amount of time gave them this heavier nucleotide, heavier isotope of nitrogen. And then you could see that, say this is the DNA molecule, okay, and this is not the final result of the experiment. This is just them standardizing the density gradient centrifugation. This is an isolated DNA molecule that they spin. And here you can see that as and when the time goes, first in 0 hour, the DNA is present throughout the tube. So this image, how do you visualize this image? Is you imagine that this is the tube. The tube is horizontal in this case. Towards your right hand side is the bottom of the tube. Towards the left hand side is the top of the tube. So here you can see in 0 hour that the DNA is present throughout the tube. As and when they start centrifugating, say in 8 hours, the DNA is present in a small region only, like the farther sides, the right and the left, the topmost and the bottommost part of the tube have no DNA. And finally, after say 43 hours of centrifugation, you can see that there is this very sharp band that is formed, and this band corresponds to the density of this DNA molecule. So here there is only one type of DNA present. So according to its molecular weight, according to its density, it concentrates towards this one region of the tube. So now you can use this and label the cells using a heavier isotope of nitrogen. So if you can see here, all these bases of DNA, whether it be AT or GC, they all have nitrogens present in them. Now, for a very small amount of time, if you give N15, N15 is the heavier isotope of nitrogen, N14 is what is abundant in nature. So if you give N15 to these cells, it will incorporate N15 in all these places of the DNA, all these bases. And this is known as a heavier DNA because its weight would be greater and because it is a heavier isotope. So if you mix N14 DNA and N15 DNA, and you subject them to the same centrifugation or density gradient centrifugation for 20 or 40 hours, you can see two sharp bands. N15 is present slightly lower in the tube because it's heavier, and N14 is present slightly towards the top of the tube because it's lighter. And there are these two very sharp bands that can be observed. So this is what Messelson and Starr claim to have taken a long time in their experiment, and the follow-up experiment is fairly simple. So what they did was they subjected the cells to N15, which is a heavier isotope, for a short amount of time, and then they removed this isotope. So at 0 hour, you can see that all the DNA present in the cell is heavy. So there is only one band, and that is the original parent molecule. Both the strands are heavy. Now, after that, after a few generations, at first cycle, there is a band that is present in intermediate of the heavy and light bands. That is, this band that we can see here corresponding to one generation is exactly in between the heavy and light. This they concluded to be because there is a semi-conservative model which we previously discussed, that the daughter strand will have one parent and one newly synthesized DNA. Now, because N15 has been removed from the cell, the newly synthesized strand will be lighter and the older strand will be heavier. So, because of this, the ultimate total density of this DNA molecule will be somewhere in between heavy and light. And that is what they observe at one generation. Then they observe that subsequently, when the E. coli is further dividing, say at around the second generation, we can see that there is an equal band at the intermediate position and a band at the lighter position. However, the band at the heavier position is completely lost. Now, this is because at the second cycle, each of these daughter strands will now synthesize in a semi-conservative manner. So the heavy strand will again serve as a template, and there will be a lighter strand that is synthesized, that will be one DNA. Similarly, here the heavy strand will serve as a template and the lighter strand will be synthesized, again the second DNA. However, these two lighter strands will also serve as templates and form these two DNA molecules in the daughter cells, because of which there is one band that is corresponding to the intermediate region because of these two DNA molecules, as we can see here, and there is one strand that is present because of the entirely light DNA that is also present. Now, these two bands are of equal intensity because it's in 1:1 ratio. However, as further cycle proceeds, say at 3, three generations, we can see that the lighter band is getting more and more stronger. And this conclusively proved that there is a semi-conservative model of replication. However, there was still one concern. Assume that there is a dispersive replication model. Here also, if you see that the daughter strand has lighter and heavier regions, that is, assuming the parent has a heavier DNA, we can see that the daughter strand has lighter and heavier regions in it. So here also, because of the similar observation, we will observe an intermediate band only. We will, and in the subsequent generation, again, it will be the same. You will observe intermediate band only. Now, to differentiate between this dispersive and semi-conservative, and to say, yes, this is what is the conclusive reason as to the mechanism in which DNA replicates, they performed another experiment wherein they took the daughter cell's DNA and you subject it to heat. Now, this will cause the strand separation. So, in this case, you can see that all of these bands in dispersive model, all of these bands should have equal heavy and equal light, causing all of these bands to show up at intermediate density. However, if you separate it in a semi-conservative model, again there is heavy density and light density, because of which there will again be the formation of these two bands that we saw corresponding to heavy and light. That is what they observed, that once you denature it, there are these two peaks formed corresponding to the two light and the heavy DNA instead of one peak at intermediate density. This is what proved that it was semi-conservative and not dispersive also. So they had three very simple conclusions that I have highlighted here. One was that the nitrogen of a DNA molecule is divided equally between two subunits, which remain intact through many generations. That is why they saw that there was always this intermediate peak, and if that gets chopped up, then the intermediate peak would have been lost. But here we can see in this experiment that there's always the presence of an intermediate peak. Following the application, each daughter molecule has received one parental subunit and the other is the newly synthesized strand. The replicative act results in molecular doubling. So at the end of DNA replication, instead of one times presence of the entire DNA pool, there is two times presence of the entire DNA pool. So they conclude that by means of a density gradient centrifugation, we observe the distribution of N15, which is heavier isotope, among molecules of bacterial DNA following the transfer of uniformly N15 substituted exponentially growing E. coli population to N14 medium. They basically say that N15 was substituted for a short amount of time, and then it was transferred to an N14 medium, and then these cells were tracked along a large number of generations. So moving on, the course in this week also spoke about a telomere and its importance. I will brief you about the importance of telomere, and then we'll discuss this very interesting paper that was recently published in Nature Cell Biology. So we saw that telomere is these ends of eukaryotic chromosomes that are basically repetitive sequences that are present towards the end and essentially form caps at the end of chromosomes. These repeats varies from one organism to another. Slight variation exists, and it protects the chromosomes from damage. The discovery of telomere and its regulation was awarded the Nobel Prize in Physiology and Medicine in the year 2009. So it's a fairly recent discovery, which was associated with aging and longevity of life. Why this actually exists is, say this is the DNA. It has this strand, which is a 5' to 3' strand. And to synthesize this, you need a primer over here. You need a primer at the 5' end. And only to that, the DNA polymerase is going to act. So this primer, once it is removed, because as we discussed, it's an RNA primer and has to be removed, the DNA polymerase cannot fill this gap. Because like we discussed, DNA polymerase has one major condition. It cannot synthesize a new strand. It can only add bases to a previously existing strand. So because of that, there would be this gap that exists. Now, in the next generation, this gap will progressively keep getting longer and longer, which will ultimately lead to the loss of or loss of DNA material from the ends. So, if this was not present, say the length of chromosomes keeps getting short, ultimately leading to a point where important information is lost from the daughter cell, and these cells will be fatal and they will not survive. So, in order to prevent this, the evolution has ensured the presence of these repeats known as telomeres. And how they were identified is fairly interesting because they observed that there is this unicellular ciliate called tetrahymena, and there were artificial mini chromosomes, like small chromosomes present within these uh, organisms. And when they were isolated and subject and like given transformed into a yeast cell, then these yeast cells would degrade these chromosomes and they were not protected. However, there were these short telomeric ends also. Once this telomeric DNA was isolated, added along with these artificial mini chromosomes, then the yeast cells retained these chromosomes and they were intact and not degraded. So this suggested that telomeric DNA had a property by which it could protect the chromosomal ends. And later, there was this other experiment wherein telomeres were artificially introduced into the cell's chromosomal ends, making sure that there's this long telomere present in the cell. And the cells were tracked across generations. So they saw that this traction of telomeres present kept getting short and ultimately reached this average length of telomeres. And after this, there was no reduction. However, on the other hand, if the telomeres were removed from these cells and there was a very short telomere present, then somehow with every increasing number of cell division, the length of telomere increased and again reached this average length and then became saturated. So this led the scientists to ask the question as to how are these telomeric ends regulated? How do the cell make sure that it always is in this particular range only? So for this, they identified this enzyme known as telomerase, which is basically a complex that is capable of synthesizing these repeats and adding them to the ends of chromosomes. So say this enzyme did not exist. Then with every cell division, the chromosome is shortened and ultimately leaves a phase wherein it is damaged and cannot replicate any further. However, if telomerase is present, then it maintains this telomeric region through many generations and replication is continuous. So how this enzyme operates is again very interesting. So this enzyme is known to have these RNA present within them. This RNA is what serves as a template, and say this is the parent strand that is incomplete because this primer has been removed from this region. Telomerase will come and with its RNA template synthesize fresh length of uh, telomeric DNA. It will increase this length. Now again a primer can be added over here and DNA polymerase can finish up this region of chromosome that is essential. So this is how the length of chromosome is maintained, and this enzyme's its structural uh, configuration is also very interesting. It has a RNA present and the end of the DNA is taken up within this hollow structure, and here it can synthesize complementary bases. Now, recently there was this paper which was entitled "An Intercellular Transfer of Telomeres Rescues T Cells from Senescence and Promotes Long-Term Immunological Memory." This is a paper that dwells into the molecular details of the transfer as well. However, we will not be discussing that. But I will introduce to you to this concept of intercellular transfer of telomeres. So if you think of immunity, right, you know that these cells have to divide for a long number of generations to ensure that your system is protected from invading pathogens. So as we saw, telomeres with every round of proliferation keeps getting smaller and smaller until it reaches this length wherein it is the cell is halted, the cell division is halted, and it attains replicative senescence. Here it's an irreversible loss of proliferation, and the cells will not be able to proliferate any further. This again is called the end replication problem. Here, what happens is, again, like I mentioned, there is this primer that is present in the lagging strand, which is removed, and this gap cannot be filled by the DNA polymerase. This will ensure that there is this fraction of DNA that is lost with every cycle of division. Now, if you think about it, there is an immune cell, and we have often heard that B cells have this clonal expansion. Now, to just briefly introduce you to this concept of adaptive immune responses, once there is a pathogen that is entering the cell, it is phagocytized by the antigen presenting cell, which will take it up inside, degrade its protein, and present it onto this MHC. This MHC has these peptides that are specific to the pathogen and can form a complex with a helper T cell. This complex formation is known as an immune synapse. Now, this APC can be of various types. It can be dendritic cells, it can be B cells. But here in this case, they have taken into account large number of cells and they've seen that this is present throughout. So a T cell also, like B cells, will have to undergo clonal expansion. And then once this immune synapse complex formation occurs, this T cell goes into this proliferative cycles and it undergoes many rounds of replication and increases its number exponentially so that it can secrete effector proteins and it can help with the activation of B cell. It can help in the synthesis of antibodies from the B cell, thereby creating this robust immune response against the pathogen. So if these if this cell did not have an active telomere, it will ultimately reach a point beyond which it will not be able to replicate. However, it is seen that in these T cells, this replicative senescence is not reached. Like they are able to clonally replicate. Today, before this paper was published, it was known that there were two major mechanisms by which telomere is elongated. One we discussed is this enzyme telomerase, and other is this recombination based method wherein the DNA loops and like recombines with itself and results in synthesis of ends of telomeres. However, in T cells, to support its replicative potential, these two mechanisms are not sufficient. So the authors evaluated whether there is something happening in the synapse only that is helping the T cells have this replicative capacity, and they saw that very interestingly, they tagged the telomeric ends of the APCs with a fluorescent probe, and they saw that after synapse formation, this fluorescence were also detected in the T cells, which meant that somehow APCs were chopping up its own telomere and sending it out in vesicles to take up for the T cells to take up and attach it to its own telomeric end. And this was very beautifully visualized in this image. If you observe the APCs, which is the antigen presenting cell, here they are tagged with EDU, which is labeled green in color over here. And the vesicles were isolated, added to the T cells, and then seen that telomeres in the T cells were tagged using FISH, which is a fluorescence in situ hybridization, which has tagged these regions, nucleic acid regions within the cell. And this was tagged using a red probe. So only if this EDU, which is obtained from the APC, and this red probe that is already present in the T cells, only if they co-localize, as in they're present together, there will be this yellow signal. And indeed, they observed that in these T cells, there was a yellow signal. which meant that in the immune synapse, previously it was not known that this was even possible between cells. And here the authors showed that the APCs were able to chop up their own telomeric ends and give it to these T cells so that they can have this unlimited proliferative capacity and not lose their chromosomal ends and not attain replicative senescence. So this is the crux of the paper. The remaining molecular details, if you are interested, we can discuss later. So that is all I have to present today. If you have any.
Excuse me, ma'am. Can you please
Yes.
explain again last slide? I didn't understand.
So telomeres are basically initially only known to be elongated by telomerase enzyme or by other recombination mechanism. There was no known records of these transfers that is happening from one cell to another. So in this experiment, which is fairly simple, what they did was they labeled the telomeric ends of the APC. So first we need to establish that the immune synapse happens between an APC, which is an antigen presenting cell, and a T cell. So they labeled the telomeres in the APC using a probe which is say green in color. Then they added it, then they allowed the immune synapse complex formation to happen. And here they labeled the T cells telomeric ends using a red probe. So say there was this transfer from the antigen presenting cell to the T cell, then only then will the T cell have this green fluorescence. Otherwise, the green fluorescence is present only in APC. And if you overlap green and red together, you will get a yellow color. And that means that both these probes are present close to each other. Now they saw in the T cell, when they zoomed in on the chromosome, there was this yellow color that was formed, which meant that somehow the telomeric ends from the APC was transported into the T cells, and here it associated along with the T cell chromosomal ends. We can see here, right? This is the chromosome, and these red and green probes are present only in the ends, which means that these are staining the telomeres.
Ma'am, these
So
these green telomeric and color were present with cell APC or T cell, T helper
it
cell.
was it is originally present only in the APC, but what happens or what these authors propose is that this APC, along with giving them the signal that there is a pathogen attack, they're also giving away their telomeric ends. So that is getting cut from the APC, and now it is present in the T cell as well.
Okay, ma'am.
This was a interesting paper that was present. However, there are a lot of molecular details in this paper that I thought
Which
would
paper
be
it
best
is? Can
to.
you please tell me?
Yes, this is a paper that was published in Nature Cell Biology. You can take a screenshot of this or view the video later. Published in September 2022.
Okay, ma'am. Thank you, ma'am.
Okay. So if we have anything else to discuss, we can. Otherwise, we can end this session.
Ma'am, how many class you conducted yet?
This is the third session. We will have 8 sessions in total. It will happen till your exams, and you can ask any questions that you
Ma'am,
have
where
till then.
we can find a recorded video version of your classes?
This will be uploaded on YouTube. The link will be sent to you all, I think through email or something.
Ma'am, already sent it or you will send?
No. They have not sent it. They will send it soon, I think.
Okay, ma'am.
Anything else that we have to discuss? Also,
So the
when the
recording
preparation is
hasn't
going.
been available yet, right?
No, I don't think it has been. The link has been sent to you people yet. They will send it within this week, hopefully.
All right. But before that, can we also get the PPTs like separately? Is that
Yes,
possible?
yes. You will get that link also, I'm assuming, because we have uploaded the PPTs from our end. So yes, that
Okay.
will also be a spoiler.
Do you have any idea when that will be?
No, I can ask them and let you know. I'm assuming within this week.
All right. Thank you so much. It would be very helpful.